In this week's lecture, we are going to discuss the anaerobes, and of course, we always focus on those organisms that are clinically or medically relevant. Now, the important concepts that you always have to think about with anaerobic bacteriology is what exactly is an anaerobe? How does it differ from the other organisms we've been talking about? What is the difference between an obligate anaerobe versus the facultative organisms? And why can't obligate anaerobes tolerate any oxygen? So if you remember way back in week 1, we talked about some of these topics where there are organisms that cannot handle any oxygen, and oxygen actually is toxic to obligate anaerobes. And that's because obligate anaerobes lack enzymes that allow them to break down some of the toxic oxidative products, so they cannot be exposed to any type of oxygen at all. Where are anaerobes found? Well, they are found endogenously, where just like some of the other organisms we've been discussing throughout the semester, we have bacteria that exist inside our bodies and all around us. So there are anaerobic organisms that are endogenous to us. They are within our bodies. We also can find anaerobes exogenously, or outside of the body, and then we can get infected with an anaerobe, just like the other organisms we've been talking about, when they get inside the body in a way that's going to allow them to cause disease. Now, the criteria for an organism to be considered an anaerobe. So in order to be an anaerobe, you have to be able to grow when there is absolutely no oxygen present. Now remember, there's microaerophilic organisms like Campylobacter. These organisms require oxygen, but they can grow minimally under anaerobic conditions. They need less amounts of oxygen. There are facultative anaerobes, and these are organisms that can grow either in the presence or absence of oxygen. And many of the organisms, especially all of our gram-negative rods, like our Enterobacteriaceae family members, are facultative organisms. And then we have our obligate anaerobes, where you absolutely can't expose these organisms at all to oxygen. They will die. Now, there are different areas in a clinical microbiology laboratory where you can incubate your cultures for them to grow. And it's very important to put your specimens in the correct environment. So there's usually what's called an ambient air incubator. This is just a standard incubator that doesn't have any modifications or gases going into it. It's just a typical biological incubator, and it contains 21% oxygen and 0.03% carbon dioxide. Now, usually microlabs will also have a CO2 incubator, and these will contain 15% oxygen and 5 to 10% carbon dioxide. Now, a lot of our specimens, if you recall, some of our respiratory organisms grow better in a CO2 atmosphere, some of our fastidious organisms. So a lot of samples you will put in the CO2 incubator, but there are others that are just perfectly fine in ambient air. Now, our microaerophilic organisms, like our Campylobacter, we have to put them in a very specific microaerophilic environment. So usually what you're going to use for that is what's called that Campy pack, and you're going to generate the microaerophilic environment within the little pouch. Now, that pouch can get put in the ambient air incubator because it's within the pouch that's going to maintain a microaerophilic environment. So as long as it's in an incubator with the appropriate temperature, it doesn't matter what kind of incubator it's in. And then there are anaerobic environments. And we will talk in this lecture about the various ways you can achieve an anaerobic environment. And in an anaerobic environment, you want 0% oxygen. So we already mentioned that there are obligate anaerobes. So there are anaerobes that might grow a little bit if there's some oxygen present, but they prefer to grow in an anaerobic environment. But then there's obligate anaerobes that absolutely cannot grow at all if there's any oxygen, even low levels present. These organisms lack, like what we said, enzymes usually to detoxify the oxygen reduction products. And those oxygen reduction products are things like hydrogen peroxide, singlet oxygen, and superoxide. These are products that are the organisms are going to be exposed to when they're inside a phagolysosome. So if our macrophage cells engulf our bacteria into a phagosome, the phagosome fuses with a lysosome. You should have learned this in immunology. And the lysosome then releases all of these oxidative and nitrosative products. Bacteria usually contain enzymes and can produce these enzymes to help them survive these types of toxic products. But anaerobes do not have these, a lot of the enzymes to help them if they were exposed to some of these oxidative products. Anaerobes also lack cytochromes that are needed to use oxygen as that terminal electron acceptor, and they contain enzymes that are active only in a reduced state in an environment without any oxygen. Now, anaerobes, like all of the other organisms that we've discussed thus far, can cause infections almost anywhere in the body. But there are areas of the body where you are more commonly going to find an anaerobic infection. And these include the head and neck area. And the common forms of anaerobic infection are abscesses, brain abscesses, sinusitis. You can get deep inner ear infections and gingivitis in the deep into the gums of the mouth. And I say deep because anaerobic infections tend to be deeper within the tissues where you're going to have a lot less oxygen present. Gastrointestinal tract infections are common with anaerobes. There are normal flora in our intestinal tract and in our mouth, in our gums, that we have anaerobes living as normal flora. So of course these organisms can cause infections. So liver abscesses, peritonitis are commonly caused by anaerobes. In the genital tract, we also have some anaerobes there as can be normal flora. So you can get genital tract infections that can cause septic abortions. And of course, our deep tissue, skin and soft tissue infections, our cellulitis, our decubitus ulcers, bite wounds, diabetic infections that get deep into the tissues. Now, like any other infections, there are risk factors where an individual might be at higher risk for getting an anaerobic infection, and these would be having a surgery or another trauma, especially trauma that's deep in the tissues. Having a poor blood supply or circulation. So diabetic patients tend to have poor circulation. So you're not getting oxygen to certain areas of the body, which of course is going to allow an anaerobe to thrive in that area. If you have tissue necrosis, so again, your tissue's dying, necrotic, you don't get oxygen in those areas, an anaerobe is going to thrive in that environment. Malignancies, and also where you have growth of other microbes in tissues. So anaerobic infections tend to be polymicrobic. So usually you're going to find multiple organisms at the site of infection when you have an anaerobic infection. So the other organisms are utilizing some of the oxygen present in killing the tissue, and then the anaerobes can start to grow as well. The virulence factors of the anaerobes are very similar to all of the other organisms we've discussed to date. So they do secrete enzymes, they have endotoxin, exotoxin, some of them can have capsules, they have spores, so they have the same types of virulence factors that we've discussed. Now, for an anaerobe, you have to be very careful about how a specimen is collected. So the list here are for your acceptable samples. If you were to get one of these samples in the laboratory and there was a request for an anaerobic culture, you can accept these and perform an anaerobic culture on them. So this would be body fluids, abscesses, usually a syringe, an aspirate from an abscess, an aspirate of deeper wounds and soft tissue infections, a transtracheal aspirate, a direct lung puncture, a suprapubic bladder aspirate. And a suprapubic bladder aspirate is when a needle is put into the bladder and then you suck urine out from the bladder. Normally sterile sites, such as cerebrospinal fluid, and also endometrial specimens. Now there are some specimens that are completely unacceptable to do an anaerobic culture. So if you were to receive one of these specimens and there was a request to perform an anaerobic culture, you would reject that sample with the reasoning that this is an unacceptable specimen for an anaerobic culture. So sputum, think about it. Sputum is coming up through the mouth, so it's being exposed to oxygen. So even if you were to have an anaerobe present, you're not going to find it, because once it gets exposed to oxygen, it likely will kill the anaerobe if it's an obligate anaerobe. Any type of exudate or materials that are collected by swabs, especially from a superficial or external wound. Nasotracheal aspirates, vaginal, cervical, and urethral secretions, and voided or catheterized urine specimens. So the only type of urine specimen you would do an anaerobic culture on would be a suprapubic urine specimen or a bladder tap where you stick the needle in. Peeing in a cup, that urine specimen would not be acceptable. It's been exposed to oxygen. So just think about that when you're thinking about acceptable and unacceptable samples for anaerobes. Has this specimen been exposed to enough oxygen in order to possibly kill any obligate anaerobes that might be present? Now, in addition to specimen collection, you have to be very cognizant of specimen transport. So you wouldn't want that specimen sitting around for too long and possibly being at risk for getting exposed to oxygen. So usually in an anaerobe, you'd like to culture it within 1 hour of collection. That might be possible if you're in a hospital setting where you are there might be a stat culture that'll come right to the laboratory after collection and you can set it up within an hour. However, if you're in a private laboratory, you're usually not getting your specimens within 1 hour of collection. In that case, if you were to receive a specimen and there was a request to do an anaerobic workup on it, you have to make sure that that specimen's been transported in an oxygen-free transport vial or tube. So these tubes contain a redox indicator. These indicators let you know if oxygen has exposed the specimen. So redox indicators commonly are resazurin, and resazurin will turn pink if it's been exposed to oxygen, and methylene blue, which turns blue when it's exposed to oxygen. So if you were to receive one of these oxygen-free transport vials and you were to see that the indicator in the vial was pink or blue, you would have to reject the specimen because the indicator shows that it's been exposed to oxygen. Now, even your oxygen-free transport vials, you should receive them within 2 hours of collection. Swabs, most of them are not acceptable unless they're in an oxygen-free transport system, and there are some anaerobic swab systems. Now, swabs should be protected from being dried out. They should be in an anaerobic system where they're not being exposed to oxygen. And whenever you receive a swab, you should receive 2 sets. You should receive one that's in the standard swab and the other that's in an oxygen-free transport swab vial, because whenever you're setting something up for anaerobic culture, you want to also set it up for aerobic, and we'll talk about that in a little bit. Now your swabs, especially if they're in the oxygen-free swab transport system, you can receive those within 2 to 3 hours of collection. We're going to talk about some of the laboratory procedures that are used for anaerobes. Now, like some of our other organisms where we've discussed doing a direct examination, you can do a direct examination on some of your specimens to look for anaerobes. So if there's a pussy or purulent appearing aspirate, if there's a lesion near a mucosal surface, if there's a presence of underlying disease, if it's an abscess or a necrotic lesion, you always have to consider if someone's been undergoing antibiotic treatment, if there's a penetrating wound like a bite wound or some sort of deep trauma. If there's gas in the tissue or the tissue is necrotic, you always want to consider an anaerobe. And if there's sulfur granules, because your actinomycete organisms tend to produce sulfur granules. So when you get a specimen in and you make a smear of it and you stain it and look under the microscope, that's what your direct examination is. You're looking for bacteria. Now, you can't usually tell if it's a facultative organism or an anaerobe just through a direct exam. You could see that there's bacteria there, maybe gram-negative rods or gram-positive cocci or something. But you don't really know what organism it is until you culture it and work it up. But these are some things that you might see that would indicate that it might be an anaerobe, and you would work it up anaerobically in addition to your typical workup. So anaerobes, one thing about anaerobes that if you have not worked in microbiology yet, you will learn, anaerobes are some of the smelliest organisms. So in the microbiology lab, when you're working up your anaerobic culture, everyone usually knows, even down the hallway, because the smell is extremely foul. So when you open up your anaerobic environment and you get hit with this strong, awful odor, you probably can say with confidence that you've got some anaerobes growing on your plates. Now, you're going to look for some of the typical colony morphology on your anaerobic plates. And I said you always do an anaerobic and aerobic workup on any specimen that requests an anaerobic workup. You always want to do both. And what you're looking for is comparing what grows up on the anaerobic plates that may not be growing up on the aerobic plates. You might gram stain some of your clinical specimens and looking for specific microscopic morphology. There is specific anaerobic media that you'll use, so you want to see if there's really good growth on a specific type of selective anaerobic media. That would be an indication that there is an anaerobe growing. If there's a double zone of hemolysis, that's another indicator that there might be an anaerobe, and these are things that you're going to look for. If there's fluorescence on another type of agar, and we'll talk about some of these agars as we talk about the organisms. The anaerobes have all the different types of gram staining and morphologies that we've been discussing throughout the semester. So you might see a gram-positive bacillus. You might see a gram-negative bacillus or a teeny tiny bacillus, a coccobacillus. You might see some bizarre shaped rods, needle-shaped, very long and slender. You can see gram-positive cocci in clusters and chains, just like we've talked before about Staphylococcus and Streptococcus. And you could possibly see sulfur granules. Now, doing anaerobic cultures, you're going to use very specific types of media, which we'll talk about now. So in order to isolate an anaerobe, you want to use an enriched media for primary isolation, just like you would for any type of clinical specimen. For our respiratory specimens, you always use blood and chocolate agar for your primary isolation media. For anaerobes, it's usually going to be Brucella blood agar, BBA. There's also Shadler's blood agar. Depending on what laboratory you're in, you might use a different type of anaerobic media, but Brucella blood agar is very commonly a primary isolation media for anaerobes. You also are going to use your selective media. Just like with our Enterobacteriaceae, we talked about hectoenteric agar and XLD agar. You also want to add in some selective media. So the selective anaerobic media are things like KV blood agar, and the KV is kanamycin vancomycin. There's PEA and PV. There's bile-containing agars. These are all selective media that are going to select for certain types of organisms. You can also use broths. So you can use thioglycolate broth, which is a nice general broth that allows a variety of organisms to grow. Aerobes, anaerobes, so thioglycolate is a nice general broth. And there's also some broths, a very rich broth called chopped meat glucose broth that some of the Clostridium organisms like to grow in. So the primary isolation media, just like always, is a non-selective and enriched media to provide optimal growth for your anaerobic bacteria. They usually contain supplements such as yeast extract, blood, vitamin K, as well as L-cysteine. And this is going to help some of the sulfur granule producing organisms to grow. For the liquid media, like thioglycolate broth, usually there's some type of oxygen binding agent in it, so that, for example, in thioglycolate broth, there's a low percentage of agar in it. So even though it's a broth, it has enough agar that at the very bottom of the tube, it's an anaerobic environment. The liquid media should also contain hemin and vitamin K, and there is something called pre-reduced media that most clinical labs use. And these media have had all of the oxygen removed, and they are actually made and sealed in an anaerobic environment. So these media, when they come in their little pouch, that is a completely anaerobic environment that those plates are sitting in. So until you break those plates open, they don't have any oxygen exposure to them. So your liquid media, we already said thioglycolate broth is a very common all-purpose media, and it's one of the few media that will also allow anaerobes to grow in addition to aerobes and facultative organisms. And then the chopped meat glucose medium, I already said our organisms, our anaerobes, the genus Clostridium likes to grow in chopped meat glucose medium. Now our selective media allows you to select for anaerobes, especially those in mixed cultures. So we already said anaerobic infections tend to be polymicrobic. So examples of some of the selective media are PEA blood agar or phenylethyl alcohol blood agar. So this agar supports both gram-positive and negative anaerobes, and it will inhibit your facultative gram-negative organisms. Paramycin vancomycin blood agar, or PV blood agar, supports gram-negative obligate anaerobes, and your egg yolk neomycin agar is very specific for the isolation of Clostridium species. We already mentioned the pre-reducing media, which most clinical labs use. This is media that has never been exposed to oxygen. It is made under anaerobic conditions. It's sealed under anaerobic conditions, and it's called PRAS, or pre-reduced anaerobically sterilized. Now, you can pre-reduce any media in the laboratory by putting it in anaerobic conditions for 24 hours before using it. But in the clinical lab, usually you're going to purchase pre-reduced anaerobically sterilized media that comes individually in these little pouches. So here is this PRAS media, and a lot of times you'll have a whole set of your primary isolation and selective media in your pouch. So you just have to open up your one pouch and you have all the media that you need in order to do an anaerobic workup on a specimen. So now we're going to move on to the second part of our anaerobe lecture.